Okay, good afternoon, everyone. If you can hear me okay, will you please type the word yes into the chat window? Okay, great, fantastic. Well, it's nice to see all of you in sort of a peculiar circumstance. I hope everyone is doing okay. Has anyone run into any difficulty or problems yet with the new coronavirus reality? Feel free to unmute your microphones if you want to speak. Ian's used to social distancing. Okay, I understand. Do any of you know anyone that's currently ill because of coronavirus? Okay, that's good. I am aware of some people that are currently in quarantine until they get their test results back. And unfortunately, one of our fellow hack students does know a person that unfortunately passed away from the coronavirus. So this is starting to get, unfortunately, all too real. Okay, so we're in the midst of some unusual circumstances here, and I would encourage you to make the right choices for yourself and your families. I will not give you a hard time at all if you are not able to attend class. As I mentioned through email, all the lectures are going to be recorded. So at the very least, when your personal schedule allows, you can go to the recordings and try to catch up on what you missed. Okay, so expect that later today, I'll be sending you an email out with some information. And that information should at the very least include a link to a YouTube link to where I'm putting the recorded lecture. So you'll get each of those every day after each lecture. Okay, before we jump into content, do you folks have any questions that you would like to ask about the revised schedule? Okay, I'm not seeing anybody making a comment on the chat window, so I'm assuming you're okay. If you're not okay, please send me an email and I'll try to get you corrected away there. It's a little uncomfortable, honestly, to be talking to my computer screen because I can't see you. I can't see your faces. I can't tell if you're frustrated. I can't tell if you're laughing. I can't tell if you misunderstand anything. So I really need you to help me know when you're not understanding something. Feel free at any moment during the lecture to unmute your microphone and ask a question if you want. If there's something that's bothering you or you don't understand, feel free to interrupt me. Also, feel free to type questions into the chat window. I will try to keep track of that to see when I need to slow down or cover something a second time. That's quite okay. Also, if any of you do have video option on your computer and you're not uncomfortable with allowing that to be on, please feel free to do so. As I said, it's not too fun talking to a computer screen. I'd very much rather see your smiling face. So that I'll leave that up to you, okay? All right, I see Sean and Hayden. That's fantastic. Folks, I hope you're doing well. Let me just mention a couple things that I think I did communicate through the email. Again, all the lectures are going to be recorded. Okay. Right now, our labs are in limbo. We were coming into a period of time with the course where all of our labs are going to be involving a lot of wet work. And by wet work, I mean DNA isolation, doing polymerase chain reaction, doing restriction digestions, a bunch of things that there is just no way in the world we can do online. So for right now, we're just going to kind of ignore the lab portion of the course and hope that our circumstances turn around so that we can get back to school. Honestly, I am skeptical that that's going to work out. As you look across the United States, you see the continual news reports of cases going up and up and up. So I am somewhat doubtful we will get back to a normal lab schedule. But just stay alert to announcements and information from me. Send me questions as you have them, and we will get through this together. Okay. So I plan on having sort of a virtual office hour after each class, starting at 2 o'clock. So from 2 to 3 o'clock, if you folks have concerns about homework or content that we're talking about, or if you just want to chat. If you just want to chat about life, about coronavirus, about things that are bothering you, that's okay. I'll be available from 2 to 3. You are not obligated to show up. You do not have to show up. It is not a requirement for the 2 to 3 o'clock office hour. That being said, this coming Thursday, I want to encourage you to try to clear your schedule so that you can be around for the 2 to 3 o'clock office hour, because there's some additional content that I think I need to cover with you. For example, you all received the grading of your last homework. It seemed to be that question 3 was a universal problem for many of you. There were parts of question 3 that all of you seemed to misinterpret. So I think it would be in our best interest if we go over question 3 from your last homework on Thursday. Also, I'm looking at the newly revised syllabus. I do not have the expectation that we're going to get any additional time for the semester, which means I need to stay on track as much as possible. That's kind of hard because we have some really meaty topics that we're dealing with and going to continue to deal with. So just put a bug in your ear that there may be times that I would like to continue some lecture conversation with you during the Thursday office hour. Okay, I'm thinking at this time that I'm going to cover DNA replication as best I can today. And then I'm going to start chapter 9 on Thursday's lecture during normal class time. And then for lab or the office hour on Thursday, I'd like to review your homework problem number 3. And if there's enough time and interest, I would like to talk to you about some recombination topics that are in chapter 7 that I don't think I can fit into today's lecture. So again, office hours are not mandatory, but I'm going to encourage you to try to be available for Thursday office hours if there's extra content we need to cover. Okay. Right before class, I sent out an email with the documents for your second literature review. So please check your email for that. And with the revised syllabus, those documents are due on April 21st. Okay, so you have several weeks to do that. All right, so a lot here. And of course, you know that your next homework assignment, the mathematical programming, the textbook chapter 6 homework problems are due this Thursday. And I'm hoping that you saw in the revised syllabus that I'm asking you to take the lab work from the DNA isolation lab that we did before break and take snapshots of your work and put that at the end of your next homework assignment so I can take a look at that. Because I have no way at this point of physically getting it from you, so we're going to have to do everything digitally. If you've already submitted that homework without including the DNA isolation standard curve and calculations, I would ask you to modify your document and please resend it to me. Okay? All right, so folks, do you have any questions right now or concerns? Feel free to unmute, feel free to type into the chat window.
So the lab that we did, the Thursday, that's going to go on our homework that's due this Thursday?
Correct. That's the only way I can get it from you, John.
Cool.
Okay. Anything else? Okay, gang. All right, so let's see what we can do here. This is a new way of teaching for me as well, so there'll probably be a few technology snags. So if you can't hear me, if I'm telling you there's a video that's being shown and you can't see it, do everything you reasonably can to interrupt me so that I don't go on for 15 minutes and you're not seeing what I want you to see. Okay. All right, so let's get back into things. We were talking about Chapter 7, DNA replication. And so what I'm going to do at this time is start sharing my screen with you. And I'm going to bring up a blank DNA Word document, and I'm going to type some notes into that document. And if the technology allows me, I might also bring up a sketch area where I'm going to try to make some very basic sketches of things that we need to talk about. Okay. So, all right, here we go. Screen. All right. So right now you should be seeing my computer screen, which has an image of a snow-covered house. All right. So all right, please give me a thumbs up in your video window if you can see the Microsoft Word document. Okay, thank you. All right, so we're good that way. Okay, I don't want to rehash too much, but there are some things, this is a difficult topic. DNA replication is a pretty involved topic. Let me remind you, and just with a couple things, I want to encourage you wherever you're viewing class at this point, that you have your textbook sitting next to you, your notebook that you write notes on, and that you actually write notes rather than just listen to me. There's really pretty good research to suggest that you will learn better if you write. So
much better if I can get familiar.
Folks, please mute your microphones so that we don't have a lot of background noise when we're not actually talking to each other. Thank you. Okay. All right, so a couple things. At the end of last class, I was trying to get started with explaining the process of DNA replication to you. So I'm just going to retype in a few ideas here. We remember from last time, we're talking about bacterial prokaryotic DNA replication. We are describing bacterial DNA replication. If I start typing too fast for you to write, please interrupt me. Remember that replication begins by unzipping the helix at the origin of replication. And you remember from last time that the origin of replication is a DNA word, right? It is a precise sequence of DNA nucleotides that is a binding pad for enzymes that help unzip the DNA helix. Okay. So let me see here. What I want to do is bring up the chat window. Let me see here. Okay. All right. I wish I had like a 70-inch computer monitor. That would help me a lot. Okay, folks. Kevin, I'm seeing you on video. Kevin, can you still see the Microsoft Word document?
Yes, I can.
Thank you. Do you see the Zoom group chat window on my screen?
I cannot now.
You cannot. Okay. Thank you. All right, gang. So right now I've brought up the chat window next to the area I'm taking notes. So as I'm taking notes, if you have questions, you can write questions into the chat window and I will see them. Okay. So, all right. So remember that DNA A protein binds first to the origin of replication. After that, DNA helicase binds to the origin of replication through its attraction to DNA A protein. Okay. Folks, all right, let's see here. All right. I think this is about where we stopped in your notes last time. Am I correct about that? Okay, Hayden is saying yes. All right. So that's basically where we finished last time. All right. So because this is a complicated process, what I'm going to do next is share a 3-minute YouTube video with you. And I will try to send you this link through an email later today so you can watch it again. But hopefully I worked very hard to try to find videos that I think really show the primary features of the process that I want you to appreciate. So I'm going to show you a 3-minute video here that goes into the process, and then we're going to come back and take notes on the video that you just saw. So please bear with me a moment while I get that video queued up. Okay. Okay. That, yes, that's fine. All right, so right now, do you see a YouTube screen up with an animation called replication fork coupling? Jose, is that a yes? Yes, okay. 1 second, gang. I need to do something to optimize the video and sound quality for you. Okay. All right, so I'm going to show you this video, and I'm going to try to make it full screen for you. Here we go.
During DNA replication, a DNA molecule is duplicated to generate 2 identical copies. The correct copying of DNA is of vital importance to the cell, as errors that are introduced into the DNA may lead to heritable mutations that could be deleterious to the cell or organism. Thus, an elaborate machinery is used both to assure correct copying and to allow the process to be regulated. Coupling of the 2 DNA polymerases, one for each strand, allows the process of fork movement to be coordinated. To understand how polymerases are coupled at a replication fork, we will first show a standard representation of the replication fork. In this typical representation, 2 DNA polymerases are moving in opposite directions, with one copying the leading strand of the DNA and the other copying the lagging strand. To expose the single-stranded DNA at the replication fork, a 6-membered ring-shaped complex called DNA helicase first separates the parental DNA strands. This separation gives the DNA polymerases the access they need to allow copying. The new DNA strand is always made in the 5' to 3' direction. Because the replication of the two DNA strands is coupled, the leading strand is copied continuously, while the lagging strand is copied in short stretches called Okazaki fragments. Each Okazaki fragment is initiated by a primase that synthesizes a short RNA primer. This primer is later elongated by DNA polymerase. The single-stranded DNA between the newly made Okazaki fragments is bound by single-stranded binding protein, or SSB. In order for the two polymerases to move together with the helicase as the fork moves, the DNA template for the lagging strand must be looped around so that both polymerases are moving in the same direction. With the polymerases now coupled, DNA replication can proceed coordinately on both the leading and the lagging strand. Once the lagging strand polymerase reaches the RNA primer from the upstream Okazaki fragment, the lagging strand loop is released, and Okazaki fragment maturation and ligation by polymerase I and DNA ligase occurs. While this maturation is occurring, the primase enzyme that is also travelling with the fork as part of the replisome initiates a new RNA primer on the lagging strand. Once this primer is made, the lagging strand polymerase on the replisome, having completed synthesis of the previous lagging strand, then re-engages with the 3' end of the new primer, forming a new loop on the lagging strand. DNA synthesis thus proceeds with the leading strand polymerase continuously engaged with the DNA, and the lagging strand polymerase engaging and disengaging as each Okazaki fragment is synthesized. To synthesize the lagging strand, the DNA template is looped out while it is coupled to the reposome complex, and the loop is released after each Okazaki fragment is completed. The length of the looped DNA at its maximal size is therefore the length of an Okazaki fragment, which in bacteria is about 1000 base pairs.
Okay, folks. First off, were you able to successfully see and
During
hear,
DNA replication,
were you able to successfully see and hear that video?
Yes, sir.
Okay, great, fantastic. Okay, so that's our foundation for moving forward here. All right. Let's see. Forgive me while I play with some buttons on my screen here. Okay. All right. If you thought my drawings in class were horrible, boy, are you in for a shocking, horrific experience at this time. Let's see what we can do here to make some sense of everything. Okay. Okay. I see. Okay. All right, gang. Sorry, this is a little bit of a technology experiment at this time. Okay. I'm going to continue trying to avoid drawing as much as possible, but there's just some processes that we really need to talk about. Okay. So new material. All right. Yeah, that's going to be a problem because that stays there. All right. I'm going to insert a new page. Okay. And I'm going to have to do something to erase that drawing. Okay. Oh, how much I miss a whiteboard at this time. Okay, so let's continue. All right, we're thinking about DNA replication. Step one. Okay, I'll just list it as step one. Please ask questions and interrupt as you need to. Okay, so remember that when the origin of replication is opened up, that's going to leave two replication forks, right? Because the bacterial chromosome is circular, okay? because the bacterial chromosome is circular, there will be 2 replication forks. So as you look in your textbook at figure 12 on page 189, figure 12 from chapter 7, page 189, appreciate that you're seeing an individual, a single replication fork with that picture. There's going to be 2 of those happening along the bacterial chromosome during DNA replication. You can kind of get a overview picture of this on figure 6 on page 184. Figure 6, page 184, you notice 2 replication forks. Okay, so I'm only going to talk about the activity happening at an individual fork, but you appreciate the activity we're describing at an individual fork like figure 12 is also simultaneously happening at the other replication fork along the circular bacterial chromosome. Okay, so doggone it, I really need to draw. Bear with me, gang. I'm going to try something here, and if it doesn't go back well, I will go back to this. All right, so okay, so all right, at this time you should see on your screen a whiteboard with 2 weird lines that resemble a replication fork. Can you see that? All right, good. Thank you for, I appreciate the heads up here because I don't want to go around if nobody's seeing anything. All right. So, okay. Let me try to do this by drawing things, and if you can't read something, you let me know and I will type it into the chat window. Okay, so what we have at this time is a helicase that's bound here. You noticed in the video the helicase was a circular molecule. It's a circular protein. It's made of 6 monomers. Okay, so over here we have this, which is helicase. Okay, it's a protein with 6 subunits. Okay. All right. So, question to everybody, you can answer in the chat window. What level of protein structure does helicase have? Remember, we talked about this in class before. There are 4 levels of protein structure. So my question is, does helicase have all 4 levels of protein structure? Yes. Okay, good. Thank you, John. Yes, it does, right? It has quaternary structure. Okay, let's continue here. All right, so remember, we've got to maintain our directionality. 3 prime, 5 prime, 5 prime, 3 prime. Okay, so let me just write some basic notes here. Number 1, DNA is unzipped by the helicase. Sorry, that looks very cartoonish. All right, the next thing that's going to happen here is that single strand binding proteins are going to attach to the single strand segments of the DNA. So everywhere we have a single strand DNA, you're going to have these things called SSBs for single strand binding proteins. Okay. SSBs bind to single strand DNA. Sorry, that's pretty bad. Single strand binding proteins bind to single strand DNA. Why is that necessary? Okay, the question becomes, ugh, what was that? Why? Why is it necessary for single strand binding proteins to bind to the DNA? Can anybody answer that for me? You can unmute or type an answer into the chat window. Okay, Hayden says to stabilize. That's a good answer. Okay, remember the physical characteristics of a single strand of DNA is like a cooked pasta noodle that's limp. So there's a very real danger that those single strands of DNA could actually flop back on top of themselves. And if they do that, they will base pair with themselves. Okay, if you have, all right, if you have a single strand of DNA, there's a real possibility that it will flop back on top of itself and base pair. And if that were to happen up here in this exposed area, you would block DNA replication. So we need the single strand binding proteins to stabilize that region to prevent them from flopping on top of each other and blocking replication. Okay. To prevent the single strand DNA from flopping onto each other, onto itself. Okay, questions, gang? Okay, here's my question to you. Where do the single strand binding proteins come from? What's the source of the SSBs? What's the source of the SSBs? Okay, here's a bigger question because I'm worried about you. What's the source of DNA A? What's the source of helicase? What's the source of the SSBs?
Do you mean what it's made out of?
How excuse me, how does it show up in the cell? Anybody?
Aren't they just proteins? So then they're being made by ribosomes, and then they're showing up in the cell just because they're being produced because replication's happening.
Okay, John, yes. And what I'm trying to drive home is that you appreciate, please mute your mic, you appreciate that that all these proteins that are facilitating this DNA replication process are themselves expressed from genes on the bacterial chromosome, right? So these things don't just magically poof into existence. You appreciate all the proteins that are allowing this process to occur have themselves been manufactured from the genes on the chromosome. Okay. All right. We can talk about that more during office hour if you like. So genes from the chromosome. Okay. All right, gang. All right, so we've unzipped the helix. We've added single-strand binding proteins. I'm not going to draw them on both strands because obviously they would be there, but since this is very archaic and I'm already artistically challenged, I won't do that. But there would be single-strand binding proteins down here as well. Okay, let me come over here and put number 3 down. What do you think is the next thing that has to happen? We've unzipped the helix. We've added single-strand binding proteins. What's the next thing that has to happen?
Synthesis
Okay, so how do we start synthesis?
with an enzyme, with a polymerase.
Okay. And what has to be attached to the DNA? What has to be attached to the DNA in order for that polymerase to do its job?
A sliding DNA clamp.
You're thinking a little bit ahead of me, and that's okay. All right, I'm seeing the answer from Ian on the chat window. Good. It's a primer. That's exactly right. So step 3, we need to add a primer. Ian, since you had the right answer, can you please type into the chat window, what is this primer made out of? Anybody can answer. Go ahead. If you know, what is that primer made out of? RNA. John, good. Okay. Okay, what is the primer made of? And it's RNA. There we go. Fantastic. So let me change colors here to green, and I'm going to put in a little squiggly here to represent an RNA primer. Remember, single-strand and binding proteins are on both strands, but I'm just not going to draw that, okay? So I'm sure your drawing looks magnificent compared to my drawing. But okay, these little green squigglies are the RNA primer. Okay, so that's the next thing you really need to pay attention to, folks, is that DNA synthesis is primed with RNA, and that should strike you as kind of peculiar and weird. Okay, DNA sorry about that. DNA synthesis is primed with RNA. Okay, so you know me, I'm going to ask you why. Why do we need to prime DNA synthesis with RNA? Why do we need to prime DNA synthesis with RNA?
Does this have to do with the 5 to 3 prime direction?
You are in the ballpark, but you're not in the infield.
Okay.
Okay. Why? Why do I need to prime DNA synthesis with RNA? Okay. I'm guessing that maybe no one knows. I'm not seeing or hearing an answer. So let me give it to you folks, okay? Remember, this whole process is happening along a circular chromosome, right? It's happening along a circular chromosome. Think about what you already know. What did we say? Synthesis occurs 5' to 3', correct? That means the 5' phosphate of the incoming nucleotide has to attach to a dangling, free, exposed 3' hydroxyl group on the growing template. If you see here at the circle, there is no exposed 3' hydroxyl because this still is a physically continuous chain of molecule that's bound as a circle. There is no exposed 3' OH group. There's no exposed 3' OH group. DNA polymerase needs an exposed 3' OH group to do its job. But here's the deal. RNA polymerase doesn't. DNA polymerase needs an exposed 3' hydroxyl. RNA polymerase does not. Am I making sense? Okay, so Caitlin's saying yes. DNA, I'm sorry, this is so horrible in terms of its appearance, but it is what it is. DNA polymerase, DNAP, can't do its job, do its function without an exposed 3' OH. But RNA polymerase can. DNA polymerase can't do its function without an exposed 3' hydroxyl, but RNA polymerase can. Because the DNA of the bacterial chromosome is still a circle, you would appreciate there's really no beginning and no end, right? If you think about a circle, there's no beginning, there's no end. That means there's no end that has an exposed 3' hydroxyl. Because of that limitation, we have to use RNA polymerase to manufacture a small piece of RNA and glue it to the exposed single-strand region of DNA. Questions? Okay, I know this is kind of intense, and as you have questions with your study time, you can bring them up, email them to me, or we can take them into the office hour on Tuesday or Thursday. Okay, so we've unzipped the helix. We've had single-stranded binding proteins. We've now attached the RNA primer, and it had to be an RNA primer because only RNA polymerase can synthesize RNA without an exposed 3' hydroxyl group. DNA polymerase can't do it. So this is one of the reasons that for people that study evolution, we actually think that RNA evolved on the planet before DNA because you actually need RNA to be used to make the DNA. Now, of course, that's not conclusive evidence, but it's one small piece of evidence that people argue is why RNA evolved before the DNA. Okay. Okay, so folks, I'm going to go to another blank screen here of white so I can start a fresh drawing. Well, let's see here, maybe. Okay. Is it okay for me to attempt to erase some of my notes here? Will that bother you? Okay, Morgan says it's okay. I'm going to try. All right, so maybe I won't have to redraw everything if I can just undo some of my notes here. Obviously, you want to do what you can do to keep things sane for yourself. Okay. What a mess we have. All right. Okay, number 4. Okay, so we've added the primer. Question to you folks. Was the primer added by, was the primer made by an RNA polymerase or DNA polymerase? RNA polymerase, exactly. We have made a piece of RNA and glued it to the DNA. You only make RNA with RNA polymerase. Very good. Okay, we'll continue here. All right. Next. Okay, the next thing that's going to happen is that DNA polymerase is going to be doing its work. Okay, so let's see here. How could I have all these colors and run out? But let's see, let me use orange here maybe. Okay, so we're going to have a new molecule of DNA added here. And what's going to do that is going to be a molecule of DNA polymerase, which I'll just draw as a square. Okay, so that is DNA polymerase, DNAP, DNA polymerase. So number 4, DNA polymerase begins extending the RNA primer. DNA polymerase begins extending the RNA primer. Okay. So let's see here. Let me ask you, okay, so Malachi, on the screen that you're looking at, look at where my little squiggly pen is. What is the directionality of this DNA molecule at this location? Should I be writing a 5' or a 3'? Please type that in, Malachi. "5' with a question mark. You're right, Malachi. Be confident. Even be confidently wrong, but be confident. Okay, so you're absolutely right. That would be 5' here and 3' here. And down here it would be 5' and 3', because we know that double-stranded segments of DNA run anti-parallel. Very good. Okay. All right. Number 6. Okay. By the way, open question to you all. E. coli, which is a bacterium, has about 5 different DNA polymerase molecules. Which DNA polymerase is the one that copies most of the DNA? Which DNA polymerase makes most of the bacterial DNA? Do you know the answer to that? Question: which DNA P makes most of The DNA. Good job, Hayden. It's DNA polymerase 3. Very nicely done. Folks, pay attention to that in your own personal note-taking time, that you know what some of these different versions of DNA polymerase are. DNA polymerase 3 is the primary major version that synthesizes most of the new DNA that will be made during bacterial cell reproduction, which you may know is called binary fission. Okay, so number 3 it is. Good. Very nice. Okay, so let's see what else we have to talk about here. Okay. All right, so open question to anybody that wants to answer it. Okay. You know from your own reading and other courses you've had that there's this thing called leading strand DNA replication and lagging strand DNA replication. Okay. Which one of these is going to be leading strand replication? Is it going to be, and so I'm going to use a fancy spotlighter here. I think I'm going to use that. Okay, right here where I have the spotlight. This molecule that's being made on the top, is it the top or the bottom that is the highlight? That is the leading strand. Which of it is it? Okay, it's the top. Very good, Hayden. Very good, Jose. Kevin, great, fantastic. Okay, all right, so that's my next question. Which new strand is the leading strand? And you correctly said in relation to our current drawing that it's the top one. How do you know that?
Because it's going in the direction as the helicase.
Fantastic. I love that answer. All right, so the leading strand is the one where the DNA is being synthesized and the DNA polymerase is moving in the same direction as the advancing helicase. You note here that this helicase molecule is moving left to right as you look at it on the screen. And you notice that DNA polymerase here where my pen is also moving left to right. So the leading strand is always the one that is being made by the DNA polymerase moving in the same direction as the helicase. Good. DNA P moves in the same direction as the helicase. And so therefore by default, the polymerase that's moving away from the helicase is going to be the lagging strand, right? In our case here, this is going to be the lagging strand. Okay. Questions right now, folks?
I had a question with the lagging strand. I know that it goes in fragments, but why does it go in fragments?
Okay, good question. Okay, let me erase a little bit more of the notes here and I'll answer your question. Excuse me if you hear some background noise. I have a telephone ringing in the other room. Okay. So Hayden has a question. Why does it happen in start-stop kind of stuttering spurts? I can answer that for you. Can anyone else answer it? Does anyone else want to try to answer it? So the question is, why does the lagging strand use Okazaki fragments? Anybody know why? Okay. Not seeing or hearing anything, so I'll write this out for you. Okay, the answer, the quick answer, Hayden, is that the lagging strand has to use the mechanism of Okazaki fragments because remember, DNA polymerase can only synthesize in the 5' to 3' direction. DNA P can only synthesize DNA in the 5' to 3' direction. Okay, so as you finish writing down that note, Hayden, have a look at your screen. I'm going to highlight something again. Okay, so look at my warm fuzzy red thing here. You notice that on the top strand, that DNA polymerase, it keeps moving in the same direction that the helicase is, which means as the helicase opens up this region of the DNA polymerase can come along and synthesize new DNA. But look what's going to happen on the bottom strand. As the helicase moves left to right and exposes new stretches of DNA here, the DNA polymerase is moving away from it, which means newly exposed segments of DNA will not get copied by DNA polymerase unless you do a start-stopping mechanism that you see in the video that I showed you. Before I say anything else to ruin that understanding, is that somewhat making sense?
Why wouldn't the loop catch that empty space?
Well, that's a good question. That's why the loop has to occur. The loop has to occur so that the DNA polymerase molecules are coupled and actually synthesized together 5' to 3'. Okay, let me back up to the video that I showed you again. Hold on one moment. Okay, hold on one moment and let me run some of this forward here. Okay. Notice that the animation video says the title is Replication Fork Coupling. That's what I'm, okay, there. Okay. Do you notice that one of these strands of DNA has to be looped so that the two green DNA polymerase enzymes can actually synthesize 5' to 3' in the same direction as the moving helicase? So the looping is necessary to physically orient one of the strands of DNA in the same orientation as the other so that both DNA polymerase enzymes can synthesize 5' to 3' and still be moving in the same direction as the helicase. Kevin, I saw a hand up. Go ahead and oh, is that your question on the chat window? Is this sort of a checkpoint to
I
ensure
was
that?
going to concur with Ian. I also cannot see the video.
Oh, I'm sorry. Okay, thank you for saying that. Hold on. Technology is magnificent. Okay, can you see the video now?
Yes, I can.
Okay, thank you. All right, sorry gang. Here we are with these troubles. Let me back up and repeat. So the video title is Replication Fork Coupling. And what that is suggesting is that the lagging strand of DNA that needs to be copied actually has to be looped so that the DNA polymerases, which are here green, can physically move in the same direction towards the moving helicase left to right on the screen. By doing that, it ensures that both enzymes can move towards the helicase while simultaneously be making new DNA 5' to 3'. If we didn't do the looping, then the green polymerase on top would actually be synthesizing 5' to 3', right to left rather than left to
With the
right.
polymerases now coupled,
I'm going to pause and let that sink in.
And there's gaps because it's not synthesizing fast enough.
There's gaps. There's gaps because, okay, there's gaps because of, hold on a sec. There's gaps because of my earlier note that as you start doing this, there's always going to be an area here that you didn't get covered because this is moving. Okay, so I really can't draw this, but this is moving. It would be looped out. Okay, this would loop this way and becoming this way, but this region is still not going to get covered. So you have to do it in loops, and each loop is about 1,000 nucleotides of DNA until the physical strain on the molecule is so much that DNA polymerase kind of falls off its template. And then a new molecule of DNA polymerase will actually hop back on here and start the same process over again to get everything that was missed by the first DNA polymerase.
Okay, I get it now.
What would help? Would it help you to watch part of the video again?
No, I just understand your explanation.
Okay, all right. I won't try to beat a dead horse, and I would encourage you to go back and review the video. I will share the link with you through email today, and really think about what you're looking at in figure 12 of page 189. That will help explain to you why the lagging strand has to loop out and has to occur in sort of chunks that are called Okazaki fragments. Okay. Sorry, this is so rough, gang. This is brutal. All right. Other questions at this time? Okay, I'm not hearing anything. I'm not seeing anything. So we will continue. All right, so we've covered this idea of leading and lagging strand, right? We've covered that. And so, you know, the last part I would really want to talk about is the termination part, okay? So the leading and lagging strands are going to be synthesized along each replication fork. And at this time, talking about bacterial chromosomal DNA replication, how many replication forks do we actually have? Can you answer that? How many replication forks do we actually have? 2, exactly, exactly. We have 2 replication forks. Remember, the replication forks are going to, can you see my whiteboard? Okay, all right, thank you. Remember, the replication forks, okay, the circular chromosome is kind of looking like this, right? And so here is one replication fork, and here is the other replication fork along the circular chromosome. The helicase on one of them is going this way around the rest of the molecule, and the helicase is going in the opposite direction around the rest of the molecule. So you have 2 replication forks moving in opposite directions around the circular chromosome, and they're going to meet up at an area of the DNA called the TER, short for termination region. You see that highlighted for you on figure 6 of your textbook. Okay, figure 6 of the textbook. So I'm going to erase a little bit of my notes here so I can put some new ideas down. Okay, so number 7 was up there. Now I'm going to put number 8 here. All right. And number 8 is simply to say that the replication forks and the DNA polymerases at each replication fork continue their work until they meet each other head on at the termination region, the TER region of the chromosome. Okay. Replication forks meet at the TER region. Okay. The TER region is going to be the same idea as the origin of replication. It is a DNA word that is palindromic, meaning it reads the same in both directions. So when both replication forks meet it, the same information content is available to both replication fork and the replication fork's pause. At this point, what we actually have is a pair of encircled DNA molecules that are interlinked like this. Kind of looks like a pair of magician's rings if you've ever been to a magic act where they've shown you the old trick of having two metal rings that are locked together and then they pull their hands apart and the rings magically disentangle. Well, obviously there's no magic happening here inside the cell, but what does happen, and let me erase some notes. What does happen is there's a special enzyme that actually can physically cut the DNA molecules apart from each other and separate them. Does anybody know from their personal study time what the name of that enzyme is called? Number 9. Okay, number 9. Question. What enzyme? Okay, I see a gyrase. Very good. That's a pretty good answer, Hayden. You are right. I'm going to give you a different term, but you're not wrong with what you said. What enzyme disentangles the DNA molecules? Okay. The technically correct answer, although I liked what you said, Hayden, is a molecule called DNA topoisomerase 4. DNA topoisomerase 4. Okay? So, gang, we're not going to go into the nitty-gritty chemical details of how it does that, but suffice it to say there is an enzyme that works at the linked areas of the molecule to cut the molecules, pull them apart from each other, and glue the cuts back together so we have two disentangled molecules of double-stranded DNA that are genetically identical copies of each other. All right. Oh, that was rough. That was rough. Gang, questions right now?
I have a question here. So I understand there's quite a wide variance of how long it really takes to complete binary fission, but in relation to the entire process, how long does the DNA replication process actually take?
A general rule of thumb, Kevin, is that most DNA polymerases, as a rule of thumb, can synthesize new DNA at about 50 nucleotides per second. So they can link in 50 new nucleotides into the template 50 times a second. So I'll let you do the math, but if you think about the E. coli chromosome, that's 4.6 million nucleotide pairs of DNA. You can start working out some basic napkin kind of calculations of how long that would take. Because remember, you have two replication forks, so you could do the calculation for one polymerase, and then divide that answer by 2, and that would give you a very rough estimate of how long it would take to at least copy the bacterial chromosome. Does that make sense?
It does. Thank you.
Okay. Other questions, gang? Okay, so I do apologize for the, you normally get crude drawings from me in class, but they're even rougher right now. So I hope you were able to follow along creating your own drawing with my speaking and talking to make some sense of things. And I am going to show you I'm going to share with you a couple video links to help you as you have your own personal study time. So at this time, I'm going to change gear. We only have about 5 minutes left, but I'm going to change gear and talk about some points of contrast for eukaryotic DNA replication, right? Everything we've talked about over the past hour has been bacterial DNA replication, but obviously we're made of eukaryotic cells, so there's some important points of distinction that you would want to think about. So I'm going to stop the whiteboard sharing, and I'm going to bring up the Word document again, so that these are just notes that I can type in for you. Okay, so I don't see anybody completely unconscious at this point. I guess that's a good thing. All right. All right. Okay. So please, if you cannot see the Word document, please unmute and tell me you can't see it. Okay. DNA replication in eukaryotes. Okay, and we're going to emphasize points of contrast to the prokaryotes. Okay, so excuse me. In the bacterial scenario that we just covered over the past hour and somewhat before break, there's a single origin of replication for several reasons. Number one, most bacterial chromosomes are relatively small compared to eukaryotic chromosomes. And secondly, eukaryotic chromosomes, because each one of those is physically larger, typically to make the process go more rapidly, there will be multiple origins of replication on individual eukaryotic chromosomes. So let me summarize it this way. Eukaryotes have multiple origins of replication, unlike the single ori in most bacteria. Okay. This is true because eukaryotic genomes are much larger than prokaryotic genomes. Think about it as an example here, right? Your human diploid genome is 6.4 billion nucleotide pairs of DNA. Okay, so that means you have chromosomes that typically have tens to hundreds of millions of nucleotide pairs of DNA that have to be copied. So to make that go faster, you're going to have multiple origins of replication. The contrast here would be for E. coli. The E. coli genome, which is a haploid genome, is 4.6 million nucleotide pairs of DNA. Another thing to keep in mind, and I know we're getting close on time, I'd ask you to please let me finish this topic. I think I need about 5 to 10 minutes, and then we will break. Eukaryotic origins of replication. Okay, so in the bacterial example, the origin of replication was defined by a DNA word that we call the origin of replication. In eukaryotic systems, in eukaryotic cells, the origin of replication is not a specific sequence of DNA nucleotides, it is a specific structure of the chromatin. Okay, so that's a distinct difference. Eukaryotic ORIs, okay, origins of replication are defined by their three-dimensional structure, not by their DNA sequence. So that's an important discussion. And frankly, there's a lot of this stuff that's really cutting edge that we still don't really truly completely understand. Okay. Number three. The human genome contains at least 14 DNA polymerases, while, as your book tells you, E. coli contains at least 5. Okay. Okay. You know, you might think, well, don't we know exactly the number of DNA polymerase genes that are in the cell? And the honest answer to that is no, we don't. Because the very definition of a gene is kind of flexible. While we certainly know most of the DNA sequence that's present, figuring out what regions of the DNA actually make up genes is a much harder question to figure out answers to. So our best computational guesses at this moment are that the human genome contains 14 DNA polymerases and E. coli contains 5. Why do the cells need multiple copies of DNA polymerases? Well, because there's different things that have to be done. For example, I didn't go over it with you, but during the bacterial DNA replication process, obviously it starts with RNA primers. While the finished DNA molecule cannot have a segment of RNA attached to it. So that means that segment of RNA primer has to be cut out by an enzyme called RNase for RNase hybrid. Don't worry about it. It's not going to be on your test. But it's going to be cut out by a molecule called RNase H, and then the exposed region of DNA that's left has to be filled in with new correct DNA, and that's typically the job of DNA polymerase 1, not DNA polymerase 3. So there's a lot of different things that make this grotesquely complicated. Okay, one more point, folks. All right. You appreciate that the bacterial example we told you was along a circular molecule of DNA. Most eukaryotic cells have linear chromosomes. So that complicates things. It makes it different. Okay. And you know from your own study time and reading, some of the homework you've been assigned in other courses, okay, that the ends of the chromosomes are called telomeres. Okay, which, as you know, have been associated with the aging process and certain diseases. The actual three-dimensional structure of the telomere region is an active area of research, and the area of the telomeres has to be copied by unique mechanisms that are different from the primary mechanism that happens at the origins of replication. So the telomeres, let me put it down in a separate bullet point here. Special mechanisms have evolved in cells to replicate the telomere region in a way that is different from the replication that happens at a typical origin of replication. As with all things that we hint about in the course, it just becomes insanely difficult. Not that it's not understandable or studyable, but we're really starting to move into the area of graduate study work where you'd have to focus specific time and energy to learn all these specific details. Okay, gang, I'm going to stop here. I'm going to go back to Zoom at this time. Well, I'll give a second for you to finish your note taking here. But we're going to wrap up here in about 30 seconds. Okay, so I'm going to stop sharing. All right. So, did you survive? Okay. Hopefully, over time, I will get a little bit better at managing some of this tech. But the unfortunate reality of it is that a lot of the ideas I want to share with you, I need to draw. I can't type it into a Word document. And so that makes it very cartoonish. So I want to encourage you over the next, starting on Thursday, if you can, to always have a pack of colored pencils or colored pens available, because it can really help life when you have different colors that you can use to design your drawings with. Okay, gang, do you have any burning questions before we wrap up here? Okay, look for an email from me later today with some additional information. I will be back online here at the same Zoom address. I'm not going to stop it. I'm going to keep this Zoom address open. I'll be back online in about 15 minutes for anybody that has a question, wants to chat. And let me see here. Morgan, if you are able, I'd appreciate if you stick around so we can work out some of those technical issues that you're having. Okay? So, gang, thanks. Be well, stay safe, and let me know how I can help you, okay? I will be back on my computer in 15 minutes. Okay.
Bye, guys.
Oh. Okay, Morgan, are you with me? Okay. So let's see here. Is there a share screen function on your Zoom session? Okay, I've never done this, but let's try having you share your screen and walk me through what troubles you're having with Mathematica and everything, okay?
Okay, can you hear me using my mic right now?
Yes, I hear you, Morgan.
Okay. Can you see what I just shared?
I'm seeing a note that says Morgan Fogg has started.
I'm sharing my Firefox right now.
Right, I don't actually see it, but it says that you're screen sharing, but I don't see your screen. What should I be seeing on your screen at this time? Oh. Uh-oh. Where did Morgan go?
She probably accidentally X'd out. I did that earlier, right before the meeting.
Okay. Well, let's hope she gets back online. Is there something that I can help you with, Hayden?
Yeah, do you think you can explain exactly what exonuclease activity is?
Exonuclease activity.
Mm-hmm.
Yes. Exonuclease activity, exonucleases are enzymes. So they're proteins that nibble on the ends of DNA or RNA. So there's two classes. You have exonucleases and endonucleases. Endonucleases will modify DNA or RNA in the middle of the chain, but exonucleases only do it on the ends of the chain.
Okay. And the exonuclease enzymes, they're just like fact checking everything that the other enzyme already did, basically.
Yes, typically they have what's called proofreading ability. You're right, fact check, yes.
Okay. But why is it opposite? Is it because it's moving this way? It said that it does it in a 3-5 direction, I'm pretty sure.
Yeah, well, when you think about our conversation, we know that DNA polymerase synthesizes 5 to 3. So if you want to proofread that, the most logical thing is to simply put your car in reverse and go backwards and check it.
Okay.
Does that make sense?
Does the whole enzyme go backwards?
That's a complicated thing to answer. I think the best thing I can say for you right now is yes.
Okay. Okay. I had some trouble understanding the recombinant duplexes. Do you think we could review that next class?
Yes. Combination is a topic that I'm hoping to do during the Thursday office session.
Okay. Cool. Okay, that's it.
Okay.
Thank you. Have a good day. Bye, Morgan.
Bye. Hi, Morgan. What happened?
I think it was my Wi-Fi.
Okay.
It was acting up in the middle of your lecture too, just saying that the connection was weak every now and then, so it might have just gave out when I decided to screen share for some reason.
Okay. When you started to screen share, all I saw was a black screen and a note that said you were screen sharing, but I didn't actually see anything.
Okay. When it gave me the option to screen share, it gave me the option to like pick out of all of the things that I had open what I wanted to share, and I clicked on Firefox. So I thought that would be the most effective way to show you. So that's what I clicked on. So I'm going to try doing that again and see what happens.
Okay. Hey,
See anything?
I see the web resizer website. Yes, I see it.
Okay, yeah, that's what I'm looking at too. So this was the picture that was for the problem 3 that I said I was able to get down to below 10 kilobytes. I think that's what it is.
Okay.
So it has the size down here.
Uh-huh.
And for what I did, I just clicked on the picture. I don't know if you can see what I'm doing, but I just clicked on it like this and dragged it into the other tabs.
Okay.
Is that what you're supposed to do when you put them into the Mathematica?
Okay, so like, so once the web resizer website resizes your image, are you downloading it to your desktop?
I do not know.
That you should do. Try downloading the new image to your desktop and then dragging it into the Mathematica.
Well, I actually do have this image already downloaded. I didn't know how to insert it without dragging it.
Okay, so do you have the file on your desktop or someplace that's easily accessible?
Yeah.
Okay.
I don't know how to, I guess I could share that, but do you know how to just explain that to me, how I could
Right.
insert
So
that?
if you want to, with Zoom, you should be able to share your screen.
Yeah, there was an option to just share the picture. Should I see if I can do that?
If you're comfortable with it, sharing the screen of your entire computer desktop might be just easier right now.
I was looking for an option to do that. I just didn't know how to. Oh, yeah, it's just the first option. Okay, desktop. Yeah, so.
Okay, yes, I see your background and everything.
Okay, so let's get rid of that. So yeah, this is it right here. This is the actual image, and then I just press browse and put that in here, and that's where it came out of.
Okay.
So from here.
Now, you have that file out open in an application of some kind, what you want to do is close it out of the application and just use the icon that represents the file and drag that icon into Mathematica.
So like if I have it on my desktop out here,
Yes.
okay, I don't have it. I don't have this particular picture as a desktop icon, I don't think.
You can use something else if you're comfortable with it.
I don't think I have a desktop icon that's small enough, though. So there might be a way to save it as the desktop. I'm not sure how to do that, though.
Okay.
I'm not computer savvy enough to know how to do that.
Okay. Why don't do you have any if you go to Google Images,
Mhm.
go ahead to go to Google Images.
Okay.
And now just type something in like Bugs Bunny. Okay, and highlight, yeah, click on one of the Bugs Bunny cartoons. Okay. And now over on the right where you have the big Bugs Bunny with the can you download that to your desktop?
Download, like in the options here.
Try that. What's it say there? Okay. When you put your cursor on the image, can you press the right button of your mouse? Is there a right button?
Let's see.
Yeah, down there it says save image as. Save image as. There you go. Now you can name it something so you know what it is. Okay, and you notice where it's saving it. It's saving it to your pictures folder. You can put it there wherever you want to put it.
Okay. Oh yeah, I'll just save the pictures. And then just save it there.
Go ahead and save that.
Okay.
Now go to your pictures folder and make sure it's there.
Okay. Let's see. I think I look for that here.
And I see Bugs Bunny down on the very edge. Do you see him?
Yeah.
Okay. So we definitely have it. Okay, so I don't want you to actually open it. Okay, so just close that window.
Okay.
Okay. But what I want you to be able to do is when you have the list of files, I want you to be able to grab that with the mouse and drag it over to Mathematica. So are you in Mathematica right now?
Yeah. I had one of them open. I just opened up the chapter 1 homework stuff.
Yeah, it doesn't matter at this point. It could be anything.
Yeah.
Just move to a blank area in your document. Okay, so we could do a couple things here. Go ahead and click the cursor in the blank area. So maybe below that last number there. Okay, all right. Now let's do this. On the Mathematica window, do you see where it says Insert? So click that and type and highlight Image. Now go find your Bugs Bunny. All right, so now it's saying drag and drop from your notebook.
That's what I kept getting.
Okay.
It kept telling me to drag and drop.
All right, that's fine. So now what you want to do is, can you resize the Mathematica window to make it about half of your screen?
Okay.
Okay. Now in the other half of your screen, we want to find the Bugs Bunny file.
Okay.
Okay.
So,
So there's Bugs Bunny. Can you highlight? Can you press with your cursor and drag it into
probably just
Mathematica?
search it from here.
Okay. Yeah. Can you drag that into Mathematica? Yeah. There. Keep going. There. That worked.
Okay.
Now, do
But
you know
will
what
it
you did?
save? So to check if it saves, I log out and just log back in, right?
Right. So go ahead and log out and sign back in and see if it's there.
Mm,
It's still thinking. There you
yep.
go.
Okay, so I can't drag it in. I have to drag in the actual, like,
Yeah, if you actually have the picture of Bugs Bunny, if you have the picture of Bugs Bunny open in an application, that won't work. You need
okay.
the icon of the file to drag
Okay.
it into Bugs Bunny.
Okay, that's what I
Drag
was
that
doing.
into. So without me talking, why don't you try it again with anything you want to try?
Okay. Let's see. So if I just searched this image I have open 8 and I drag that in right here. No, it's really small now. Sign out. Okay, yeah, it's still there.
Okay, very good, very good. Okay, so that works.
Yeah, so that should solve it then.
Okay. Now your web resizer, like if you take a snapshot with your iPhone camera, how have you solved the problem that your picture sizes were so large that even web resizer wouldn't work, or that you couldn't email them to yourself or something like that?
Well, since I have that cap on my email where it won't let me send emails of a certain size, I've just been airdropping them to my computer since it's a Mac. And my phone's an Apple. They can communicate with each other that way, so I've just been airdropping. And that works quicker than email, actually. So I still
Okay.
have that option.
That's fine. If that's working for you, that's okay. Can you please try that the picture of your notes that you just put in the document? Can you select it and try to drag it and make it bigger?
Yeah. Like this?
Yes.
Mhm.
Okay, good. I just want to be sure that you're not creating files that are so small that I lose the quality of the image that I can't read your notes, but I can read that. I can read
Yeah.
that.
And this is really small. This is like 8 kilobytes.
Okay, so that would be fine.
Mhm.
The only other things I would mention is when you drag a picture file of any kind into Mathematica, be careful that you give Mathematica enough time to save that new information before you log out of your Mathematica document. Because,
Okay.
you know, if you had a picture file, let's say that was 100 kilobytes, which is not big, but it's still pretty big.
Mm-hmm.
If you drag that into Mathematica and you said, oh, I'm done, and you close Mathematica before Mathematica could literally save all that new information, then you will miss it.
Yeah.
So do you see in the upper left-hand corner of the Mathematica document where you see the name of your file? It says Morgan Fogg Homework. You see
Yeah.
that?
Mm-hmm.
If Mathematica is still saving information, you're going to see a little icon up there that's kind of moving or circling around, letting you know that it's still doing something. And if it's doing something, you really don't want to close your document at that time.
Okay.
Okay?
Like that.
Yes, exactly.
Okay.
Exactly. Okay. So does that solve your main issue?
Yeah, it should. It should.
Okay. All right.
Mm-hmm. Thank
Anything
you.
else? Anything else I
No.
can help you with?
No, that was my biggest issue. I don't have any other questions right now.
Okay, Morgan. Thanks for hanging around, and I'll see you on Thursday, okay?
Yeah, thank you so much for helping me out.
Sure thing.
Bye.
Bye-bye.